Bonjour, hello, and kia ora koutou katoa. My name is Ange McGaughran. It's not a straightforward one to pronounce, and it doesn't get pronounced well in New Zealand either, so don't worry about that at all. It's a pleasure to talk to you today about some of the work that I did when I was based in Australia before I moved home to New Zealand a few years ago. Okay.
Doesn't work. You have to click back on the slide, I think. Une fois si tu as enlevé le petit panneau, du coup, voilà, il faut recliquer sur voilà.
Okay.
Là, ça doit être...
Now, yeah, okay. Thank you. So for the next sort of 30 minutes or so, I'm going to tell you a bit of a story about my favourite pest moth at that time, Helicobirpa armidra. I'll start off by giving you an introduction to myself and then also to the system. And then I'll talk you through three separate little stories about focusing on different elements of this pest moth. So first up, introductions. So as I said, my name is Ange, and I originally I've always sort of focused on invertebrates through my career, just sort of by chance and serendipity. When I started off, I did my PhD focusing on springtails that lived in Antarctica. And after that, I moved to Germany, where I spent 4 years and focused on this nematode that lived underneath the wings of a beetle. So it was a very interesting necromantic relationship that I was looking at there. Then in 2014, I moved to Australia, and this is where I first started focusing on Helicobirpa armidra, where I was using museum samples of this moth, and that's what I'll talk about today. I ended up spending quite some years working on this system, first off at CSIRO and then across the road at the Australian National University. And then finally, I moved back home in 2020, right before COVID lockdown, and started working on my new favourite system, which is blowflies. And you can see an endemic species pictured just there. So in my lab, which is called the Invasomics lab, we focus, based on the name, hopefully you can tell, on the genomics of invasion, of biological invasion. And what we do is we use a variety of different techniques to look at and to try and understand what makes an invader a successful invader. So what kind of traits and genomic signatures enable invasion success. And we do that predominantly using next generation sequencing, but we also have a rearing facility and ask a lot of ecological questions about these different blowfly species. But today I'm going to focus on Helicobirpa armidra, as I mentioned already. And as you may or may not know, this is a major global pest that causes billions of dollars in crop losses and management costs globally. And you can see the variety of different crops that it feeds on pictured down the bottom there. It's highly polyphagous, so it feeds on over 300 different types of crops. It's got a large, diverse set of mechanisms internally that it can use to detoxify chemicals in plants and respond to environmental cues. And it's also highly motile, so it's got a pretty widespread global distribution, including a relatively recent incursion into South America in 2013, and tracking northwards to represent a real threat to the cotton industry in North America as well. And so what that means altogether is that novel genetic changes can really rapidly sweep through populations. And in particular, insecticide resistance is something that can really rapidly permeate through populations as individuals are moving and exchanging genes freely. And this is one example that sort of shows you how the species has become such a successful pest. So what you can see here is a phylogeny of a group of, we've got Helicobirpa armidra and its sister species, Helicobirpa zaya, up here. And you can see that they're polyphagous, and then the rest of these groups, as we get further and further out, either have one family of species that they feed on, one crop family, or one species or genus. So the bluer colours become more specialised to specific crops. And what you can see pictured on the right here different gene families, and are coloured here by these different by this key. So we've got gustatory receptors, GSTs, CCEs, and cytochrome P450s. And what's really interesting about those gene classes is that they're responsible for detoxifying things like like insecticides, and also for helping process food through the gut. And what you can see is that the more polyphagous groups have got these expansions in these gustatory receptors in particular, and also some of the other gene families as well. So this is one of the key genetic mechanisms by which these species become really good at being pests. And I was sort of interested in examining that question further from a genomic perspective to understand how they become so successful. But first I wanted to understand the work that had already been done in the species with respect to genetics. So I'm going to spend a little bit of time going into the details of that. So what you can see pictured here is phylogeny. Well, it's a haplotype network based on mitochondrial data. And you can see that individuals are coloured from the location in which they were collected. Over here on the left, in the black and the white, are predominantly Australasian samples from New Zealand and Australia, but there are also some of those samples here and here in the tree. So the general consensus is that there's no global population structure. There's a lot of free gene flow being exchanged even across the globe for this pest. And this is just borne out again in a different representation, this PCA plot on the right. And then if we look now at genome-wide single nucleotide polymorphism or SNP data, then again we've got this PCA type structure. Up here we now have Helicoverpa zea represented on the plot in the grey, and this is the Helicoverpa midra set of samples. Over here we have the full kind of global populations, and then over here on the right we have these Australasian samples. So they're falling out a little bit more differently in the SNP data. And what that ultimately corresponds to is the potential presence of these two subspecies, Helicoverpa midra converta, which is present in Australasia, according to this study, and then the rest of the world, which is Helicoverpa midra, which corresponds to all of the other populations in the study. But what you can see, even as we look at this particular ancestry plot, is that this converter group still retains a lot of shared ancestry with the rest of the world. And so I've put this sort of question mark here because although this study sort of provides support for this subspecies structure, it only had samples from Australia from a very restricted geographic distribution. And so one of the things I was interested in looking at was whether or not we would still see this subspecies structure once we had samples from a wider geographic distribution in Australasia. So the design of the project that I worked on, various aspects of while I was in Australia, used over 260 samples collected from across Australia, and you can see the sample number within each state plotted there on the figure. And what this corresponded to as well was museum specimens that spanned over 100 years in time as well. And so what you can see plotted here is on the left-hand side, samples that were collected prior to 1950 corresponded to a time when insecticides were not being used in Australia. And then I have samples that spanned through the decades right up until the sort of early 2000s. And this corresponded to the use of different insecticides through time. And what you can sort of see plotted here is on the left of each thing is when the insecticide was first used in Australia. And then on the right-hand side is the first noted resistance to that insecticide in the field. And so, for example, if we look down the bottom at DDT, it was first introduced in the early 1960s. Resistance was noted in 1975, and then that actually ended up being banned in Australia in 1987, and then subsequent other types of insecticides were used through time. So the experimental design has a spread of these samples through both time and insecticide classes being used. And what I used predominantly was a method known as targeted exon capture. And this is a method where you design a bunch of baits, a bunch of areas of the genome that you're particularly interested in capturing. And so what I did was I designed that around genes that I expected to be relevant to environmental sensation and tolerance, and also insecticide resistance. And so I pulled these particular regions of the genome out of those museum samples and examined them through time. And not that you should really read this in depth, but these were all the different gene families that I looked at. And the main thing to take note of really is that corresponds to functions like digestion, host use, environmental sensation intolerance, and detoxification and insecticide resistance. So these were the genes that I was particularly interested in examining. And so the problems that I wanted to solve across the years that I was focusing on this project, in the wet lab, I was interested in looking at some of the tricky aspects of dealing with museum samples that are often low in quality, low concentrations of DNA, and I wanted to understand how the age of the sample affected my ability to get high quality data back. Then in two purely more bioinformatic projects, I focused on answering that question around how connected Australian populations are to the rest of the world and whether or not we found support for those subspecies with the additional Australian samples. And then, of course, the most interesting question, I think, was how rapid evolution proceeds mechanistically and whether we could find signatures in the genome that corresponded to selection operating really heavily in response and responses at the genomic level to those different selective pressures provided by insecticide use through time. Okay, so the first focus on the wet lab around how sample age affects data quality. I've put up, along the way, I've kind of put up the details of software and things like that, which I won't focus on too much, but those details are there and the slides will be shared later and you can ask questions at the end if you have any questions. But a key thing to note here, just because of the age of the samples, is that it was a bit tricky getting DNA out of these old samples. And so a couple of modifications to the protocol included using this user enzyme, the special enzyme that can extract out uracil bases, which are a common signature in degraded samples. So that was something that I added to the protocol, and I'll discuss whether I think that worked in a sec. And then I used a variety of different sort of software to look at the damage in the genome. So we expect to see a higher frequency of C to T and G to A transitions at the ends of the reads if they're highly damaged or not, for example. And then I looked at a whole bunch of different analyses, relationships between sample age and these different metrics of data quality. So this is the first look at how the effects of sample age were detectable at the genome level by looking at these C to T transitions and the G to A substitutions. And so, as I mentioned, you sort of expect to see a region of these increasing in frequency at the beginning and ends of reads, and there's no such signature, no appreciable signature for that. So it looks like that user enzyme worked quite well at getting rid of, chopping out those bases and shortening the reads at those sites. And then down the bottom, I was looking here at whether or not the frequency of those transitions at the first base pair, so right at the beginning of the reads, whether that was correlated with sample age or not. And you can see that there's no particularly strong relationship there. So it seemed that sample age and that damage signature was not an issue in these samples. And then one of the other things that I was interested in is if you are familiar at all with the kind of library protocols associated with dealing with older samples, then we typically start off with very low concentrations and we have to use PCR to amplify up the amount of DNA that we have. And as soon as you start doing that, you introduce additional artifacts into your sequencing data. So in this case, what I could see was that older samples definitely had these lower starting concentrations of DNA and required more PCR cycles. And so as a result of that, what we ended up getting was that this is a whole bunch of different kind of metrics of sequencing quality, which you don't need to pay too much attention to, but the idea overall is that the younger samples had better quality, as you would expect. So as samples are older, they become lower in quality. And as a result of having to do these additional PCR amplification cycles, what ends up happening is that as well as the DNA sequence, you also amplify a part of a short sequence known as an adapter, which is what you use to tag your sequences with. And so older samples ended up having more of the sequencing data that came back was contaminated with this adapter signal. And in the worst cases, up to 82% of the sequencing data that we returned back was actually just adapter sequence. And I bring this up in particular because when you're working with exon capture data, it's such a small region of the total genome that you're recovering. I think in total my capture size was around 3 meg. Which is very small, 3 million base pairs, as opposed to like a whole genome. So actually having only 18% of the run correspond to DNA sequence, it still actually returns quite a significant volume of usable data, even though that looks like a really bad metric. This was true, this sort of negative relationship was true whether I mapped my reads back to the whole genome or just to that 3 million base pairs that I was trying to capture. And another metric that was sort of interesting was this measure of saturation, which is an indication of whether sequencing more will actually give you more breadth of data. So not more depth, but actually covering more breadth of the genome or not. And it turned out that this was not the case. So there was this negative relationship with sample age there. So the older the sample, you don't necessarily want to do more sequencing, or you're just going to get more adapter, essentially. So from this particular part of the study, the key kind of take-homes are that, of course, as expected, age has got a really big impact on data quality, a really strong negative relationship. However, using something like this user enzyme to get rid of that degradation damage can be a useful approach. And since I started this project, there are now these dimer-free methods that guarantee you in the library prep process that you don't actually end up with that adapter contamination, which is really nice. So there's been some advances in the sequencing technology and associated library prep protocols that really work well for museum samples now. And there's more information in this publication if you're interested in delving into the details of that. So the second part of the talk that I want to focus on now is that first question around population structure and how connected Australian populations are. And the other thing I was interested in asking for this part of the study was whether there's any value in the bycatch. So you might remember that I said that I sampled and I particularly pulled down these particular regions that I was interested in. But what happens when you do exon capture is you always get some off-target sequencing as well. So you get some sequences that weren't in your bait design and you're sort of paying to sequence them as well. So what I wanted to see is whether I could actually get any value from that bycatch data. And in this case, quite often what people will do when they look at bycatches, they'll look at mitochondrial regions because they're present in a higher frequency in the cell. And so often the off-target sites are mitochondrial. So what I did was I used the same data from the previous study, around 260 samples, and then I combined them with 53 whole genome resequencing data samples that were available from a previous publication. And as I mentioned before, we, with this study, are substantially increasing the geographic coverage compared to this previous work that had focused really just on sort of one region in New South Wales. So this covered now, the study now covers all of the Australian states except Tasmania, and then a whole bunch of other sites from this study as well across the world. So that's a little bit of the sort of methodology, but the important thing is that what I was interested in looking at was not just can we get sort of usable information from the bycatch, but also how does coverage of the bycatch impact our results? So what I did was I subset the data set into different degrees of coverage. So I retrieved the information and then I artificially created only 5% coverage, only 10% coverage, right up to 65% coverage, so that I could see across the whole mitochondrial genome, so that's about 16,000 base pairs, what kind of percentage of coverage do we really need across that breadth in order to make robust evolutionary inferences. And in addition to the mitogenome data set, which is this part, I also gathered over 800 location tagged CO1 sequences from GenBank to have a look and see whether the results that we were producing from this missing data mitochondrial whole mitogenome data set would match up to what was in the public databases for just the CO1 gene. So here we performed sort of a structuring analysis to explicitly test for that presence of the two different subspecies, so the Amydra and the Amydra converter. And then we also had a look at this phylogenetic analysis using these Bayesian skyline plots to have a look at changes in population size. So two just sort of fairly common population genetic techniques, and looking to see if the results that we got across the different coverage data sets were consistent. So rather than sort of a little bit of questions about sort of the evolutionary inference itself, but more so how much that inference might change with missing data. And so this work was performed predominantly the analyses by Leo Featherstone, who's now at the University of Melbourne. And the first plots that you can see up the top here are just really looking at the relationship between the proportion of the mitochondrial genome that's covered on this axis here, and then the sample age. And over here, I've put, instead of the sample age, the file size, because theoretically the older samples are lower quality and they should have a lower sample size. But what you can see is that there's no real strong relationship here in terms of the amount of coverage that we were able to retrieve and the actual sample age, which is kind of interesting. Probably this is because the mitochondrial genome is there and is well represented even in older samples. And then down the bottom here, you can see these coverage heat maps for the 5, 10, 15, and so on, different coverage mitochondrial whole mitogenome data sets, and then on the right-hand side, the CO1 data set. And if it's coloured in blue, that means that it's covered, the base was present, and if it's grey, then it's absent. And individuals are plotted in random order down this axis here. And so what you can really see with this is that there was no particular huge gappy part of the genome. So there wasn't a huge area of the mitogenome that wasn't captured for some reason, like a high AT content or something like that. And so this first question was around subspecies support. And so here we've got the results from the 5% through to the 65%, and here's the CO1 data set here again. And you can't really see the colours too well on this plot, I'm afraid, but it's coloured basically with either sort of a darker blue colour to support the converter cluster and a red colour to support the Amydra cluster. And what I was particularly interested in really was whether or not we could find support for the Amydra, so that's the rest of world cluster, on the Australian continent, because the previous research had really indicated that Australasia was the converter region. And you can see that there was some support for this distinct grouping. So we found support for two distinct clusters, but there wasn't sort of a geographic turnover. The Australasian subspecies is present in the rest of the world, and the rest of the world species were present on the Australian continent as well. And on the right-hand side, what we've got plotted here is the proportion. So you might remember that the data set includes both rest of world individuals, so both converter and Amydra individuals, the two subspecies. And so on this one, we've got the proportion of the Amydra subspecies, so that's the rest of world samples in the whole data set. So that's the proportion on this axis here. And then on this axis, we've got the degree to which the DAPC analysis supported admixture, so more than a non-binary signal. And so what you can see here is that the admixture portion is increasing as the proportion of Amydra. So as we get a greater proportion of global samples in the data set, then we start to see a significant effects on admixture proportion. And so it's not just the actual coverage that's important. So our coverage is indicated here on each of these dots. It's not just the degree of coverage across the mitogenome, but also the composition of the data set that's important in driving some of the signals. And then the CO1 data set, which is over here, shows a contrasting pattern in which it had a higher proportion of rest of world samples, because I had downloaded all these samples off of GenBank. But it showed this lower pattern of admixture, even though it's, it should sort of be somewhere up here, but it's actually over here. So what this really tells us, the kind of take-home message from this part, is that The sampling bias by itself doesn't explain that signal of admixture, and the higher coverage that we have in the CO1 data set allows that clearer separation of both into both of the different subspecies. And then this part of the study was just to have a look at some common kind of demographic analysis to see how this changed, how our results changed with the increasing coverage of the data set. So again, we've got 5, 10, 15 and so on, up to 65%. And what you can see here, this is looking at population size change through time. And basically, we've got present day sort of on the right-hand side, and all of the plots sort of show this continual increase in population size for each of the data sets. But what's interesting is that if we look at this top set of data sets, you can see that the age, so the span of the x-axis goes back a lot further. So this one here, we've only gone back to sort of 1700, and in this one here, it's going back past the other side. So it's just this tiny part here that's reflected in this data set. So what that means is that when we have lower coverage, so we have more missing sites in the data set, the software infers an older divergence history. So it's sort of making up for that lower coverage by inferring like that it's more an older divergence event. So some key points from this part are that mitogenomes assembled from bycatch with up to as much as 75% missing data give us robust evolutionary inferences. But when we are in that lower coverage range of sort of 5 to 20% coverage only, so the predominantly missing data coverage sets, then we start to see some of our inferences starting to become less robust. But it wasn't just the data set coverage, but also the composition. So the more we had a wider geographic distribution in our data set, the more robust our inferences were in terms of being consistent with the broader literature. So really this study was kind of demonstrating the value of museum samples as important records of historical change through that bycatch, because sometimes people have a particular question in mind that they're interested in asking, and there's this additional kind of resource that can be tapped into via that bycatch. But I guess the caution really is to sort of check your coverage before you start making too many inferences, and make sure that it's decent, at least over 20%, probably more, so that confounding factors aren't really driving the overall inference. Okay, and then for the final few minutes, I'm just going to focus on the last bioinformatic question, which was around how rapid evolution proceeds at a mechanistic level. And so this is some work that was led in recent years by Andy at CSIRO and Ali, who is now actually at the University of Auckland. Okay, and so again, I'm reusing that data set that I described in the previous study, but now what I've done is I've subset the data into decades of time, and I'm looking for temporal patterns in the data set. And in this case, I'm looking at just the SNP data from those different exons. So first up, interested again in kind of looking at this question around population structure, but here it was more about, is it safe to sort of combine these samples together and look at these inferences across time, or is there a really strong signal within a particular decade that suggests that it's not really a reasonable thing to combine them? So this particular analysis over here shows us a PCA plot showing the kind of population structure through time. So again, you probably can't quite make out the signal, so you just have to trust what I tell you. Essentially, there's no population structure corresponding to time, although we do have the small number of samples here from the 1970s. They are also present in this side of the plot, but there's a small subset of samples from the 1970s that were collected in Western Australia that fall out a little bit differently. It's not a huge amount of percent variation. But there may be something interesting that was happening in Western Australia in the 1970s, so that's something that could be looked at further. But the sort of admixture plots gave us two main clusters, which you can see pictured in purple and green. And the admixture sort of occurring within decades is relatively consistent. There's no absence of admixture in the pre-1950s before insecticides were used, but so admixture being a signal of sort of gene flow events happening. And what you can also see is that there does seem to be a bit of an increase in admixture in the 1960s and compared to the 1970s. So there's sort of some interesting gene flow exchange things happening in the 1960s. And then it seems to drop off in the 1970s and then pick up again a bit later on. And what's interesting, what you can see plotted across here is the use of these main insecticides. So here's DDT, perhaps having a strong impact here in the 1960s. And then over here, we've sort of got synthetic pyrethroids potentially having an impact later as they're passing through populations. And then what I was particularly interested in looking at was understanding signatures of selection through time in this system. So I used a method called PC Adapt to look at outlier SNPs. So these are SNPs that are different to the kind of baseline signal and are good potential candidates for being under selection. And the first thing I did was just look at the proportion of those outliers through time to see if there were any changes. And so on the left-hand side here, you can see the total number of outliers and how they change through time. And what you can see is that there's an increase here in the 1960s and the 1980s relative to the rest of the time periods. And then on the right-hand side, I plotted the same information, but by gene family to see if there were any sort of interesting patterns or things that stood out. And again, you can see the same pattern by which the 1960s and the 1980s, which corresponds to the time when, as I said before, DDT and synthetic pyrethroids were being heavily used around that time. So something interesting happening. The next thing that I wanted to look at in a bit more detail was look at these outliers and see how their frequency in the populations was changing through time. And so that's what you can see plotted here. So on the left-hand side, I'm comparing the pre-1950s, so before insecticides were used, to the 1960s versus the 1980s. And on the right-hand side, it's to the 1980s and the 1990s. And so what I'm asking is a particular allele, a particular outlier at a particular frequency in the 1950s, how does that change as I move to the 1960s and then to the 1980s? And there are a few different possible options. Either there's no change in that allele frequency at all, which is happening at the predominant number of sites. There's the opportunity that the allele frequency alternated, so it changed, but it went up and then down, or it went down and then up. And then there's the possibility that it just went up and up as we moved across those 3 time points, or down and down. And so what you can see is that up to 50% of the outlier allele frequencies changed from one period to the next, and that's across the different gene families. And the changes, when they did change, were mostly alternating. So they mostly went up and then down, or down and then up. And that's very suggestive of a process like antagonistic pleiotropy happening, whereby a selective pressure makes it beneficial for an allele to change in frequency. And then in the absence of that selective pressure, so perhaps that pesticide or an insecticide is no longer being used anymore and a new one is, there's a fitness cost for maintaining the original allele at high frequency. And so then it very rapidly drops back to low frequency. So that's a really interesting hypothesis that we have taken out of this work. The other thing that I was really interested in doing was looking at the actual genes themselves, the interesting genes. So we used this program called SNP Effect to have a look at the impact of the different outlier alleles, and to see whether those changes were, deleterious, had high, low, moderate impact. Most of them had low deleterious effects across the different gene families, as you can see pictured here. And then the next thing that we did was we were interested in trying to understand, basically looking at sort of these changes through time in a slightly different way to the way that I looked at them before. So first of all, we filtered out any alleles, outlier alleles that had low support. And then we calculated the mean non-synonymous to synonymous variance per gene. So changes in alleles that either change the amino acid or don't change the amino acid. And this is sort of similar to looking at a Ka-Ks ratio, if you're familiar with that terminology. But here, instead of looking at sort of changes across species, we're looking at changes within species, but through time. So we sort of tweaked the way that we did it. So we use this method to get a sort of a base rate. and then per gene, and then we compared that to that same ratio, but this time over time. So anything that was changing in this one relative to that one, we were interested in. And what you could see is that most of the changes were very low. Most of the genes showed no change or very little change across this ratio, but a subset of them showed more than twofold change through time across at least one sequential decade. And those ones we were interested in looking at further. There were 21 of them, and they corresponded to 8 different gene families, as you can see pictured here. And CRPs, which is our chemosensory proteins, so important for sensing the environment, had the highest number of outliers in this gene. And these highest number of outlier changes through time. So the key points for this part are that there was no clear population structure within Australia, no signal for kind of geographic turnover between those two subspecies. The highest outliers that we saw were in the 60s and the 80s, which corresponded to the use of those key, really intensely used insecticides sort of before integrated pest management plans came along. We found major outlier allele frequency changes through time and potential support for this antagonistic pleiotropy. And then, of course, there were 21 interesting genes with these major shifts in allele frequency through time, and they likely have a function in resistance. And with that, I would like to thank folks at the Australian National University in Cairo, which is where I did the work, being hosted by Craig Moritz, pictured here. The museums across Australia that let me sample their precious samples, collaborators listed here and mentioned as we went, and then some funders for the work while I was based in Australia. And thank you for your attention, and these are my contact details as well.